Inheritance patterns are the different ways in which traits are passed from one generation to another. Inheritance relies on homologous chromosomes, which come in pairs, one from mum and one from dad. Each chromosome has genes, which are segments of DNA that carry genetic information for a specific trait, and different versions of the same gene are called alleles. As an example, brown eye colour and blue eye colour are both alleles for the eye colour gene, and each parent offers one allele of a gene. Now, these alleles can be either dominant, often represented with a capital letter, or recessive, represented with the corresponding lowercase letter. The difference being that it only takes one dominant allele for its traits to be expressed, whereas it takes two recessive alleles for its traits to be expressed. Human somatic cells, that is, all of the cells aside from the sperm and the eggs, which are called gametes, have 23 pairs of chromosomes, 22 somatic pairs and one sexual pair, adding up to 46 chromosomes in total. For the sex chromosomes, a genetic female has 2 X chromosomes, while a genetic male has an X and a Y chromosome. All 46 of these chromosomes, along with the alleles they carry, segregate during meiosis, which is the process of making gametes. Gametes only carry half the genetic information of the parent, so 23 chromosomes. Females require an egg and a sperm that are both 22X, whereas males require an egg that's 22X and a sperm that's 22Y. Once the male and the female gametes merge during fertilization, their alleles combine to give rise to one of 3 possible genotypes of the offspring: homozygous dominant, or big A, heterozygous, or big A, little a, and homozygous recessive, or little a. This genotype determines a person's features, or phenotype, such as hair colour or whether or not they have a genetic disease. Genetic diseases develop when a gene doesn't work well because of a mutation that affects one of the 2 alleles, and if the person has children, these mutations can be inherited. To get a quick picture of how different inheritance patterns work, we'll use a pedigree, where we represent females with a circle and males with a square. We'll shade in the individuals with the disease, so it's based on phenotype, not genotype. When a mutation affects a dominant allele, it only takes one mutant copy to cause a disease. This is a dominant inheritance pattern. However, if a mutation affects a recessive allele, it takes 2 mutant copies to cause a disease. This is a recessive inheritance pattern. Now, when the mutant allele is on a somatic chromosome, it's called autosomal inheritance, and when it's on a sex chromosome, it's called sexual inheritance. Sexual inheritance is divided into X-linked inheritance, where the mutant allele is on the X chromosome, and Y-linked inheritance, where the mutant allele is on the Y chromosome. Finally, there's mitochondrial inheritance. In addition to the 46 chromosomes in the nucleus, cells also have mitochondria which carry their own DNA. When an egg and sperm come together, the cytoplasm and organelles like mitochondria come from the egg. So when looking at the pedigree of a family affected by mitochondrial disease, you can see that, as a result, both males and females can develop mitochondrial diseases, but only females can pass those diseases on to their children. So let's look at autosomal dominant inheritance. An example would be Huntington's disease, where there's a mutation resulting in a dominant allele on an autosome. So both homozygous dominant people, big D, and heterozygotes, big D, little d, have the disease. But homozygous dominant people rarely reproduce because the disease is too severe. So most of the individuals that reproduce are heterozygotes. So let's say that we have an individual who's heterozygous, or big D, little d, and that the other parent doesn't have the disease, so they're homozygous, little d. Since the mutation is on an autosome, it doesn't matter which parent has the disease. Now let's create a Punnett square. The affected parent can make either mutant gametes, which carry the big D allele, or normal gametes, which carry the little d allele. The healthy parent only makes normal gametes carrying the little d allele. When these gametes combine, the children have a 50% chance of being heterozygous, big D, little d, and having the disease, and a 50% chance of being homozygous, recessive, little d, and not having the disease. In a family pedigree, the inheritance pattern would look like this, with affected members in every generation. Usually a person with the disease has one affected grandparent, affected and unaffected uncles and aunts, one affected parent, and even affected and unaffected siblings. In addition, the mutation can also appear spontaneously in a person whose family isn't affected. Now, autosomal recessive diseases, like cystic fibrosis, only occur when a person has 2 recessive alleles, or r. and heterozygotes with only one mutant allele, or big R little r, are considered to be carriers of the disease, even though they don't have the disease. For example, if two parents are carriers for cystic fibrosis, they are both heterozygous, big R little r. Let's use a Punnett square again. They make gametes that have the normal big R allele and the recessive mutant R allele. So their children have a 25% chance of ending up homozygous dominant, or big R, which means that they don't have cystic fibrosis, aren't carriers, and so are healthy. They have a 25% chance of ending up homozygous recessive, little r, which means that they do have cystic fibrosis. And finally, they have a 50% chance of ending up being heterozygous carriers, big R, just like their parents. However, if one of the parents is affected, little r, and one of them is not, big R, then all of the children will end up heterozygous, big R, and they'll be carriers. So in a pedigree, the pattern is that the disease tends to skip generations, so a person with the disease usually has unaffected parents, or even grandparents, but they do have affected great-grandparents who passed on the recessive mutant allele. So, members of the same family have a higher chance of having heterozygous genotypes compared to the general population. That's why children of consanguineous unions, where two related individuals have children, are more likely to have autosomal recessive diseases. Now let's switch gears and look at sex-linked inheritance patterns. Males have only one copy of each sex chromosome, meaning that they have only one allele for the genes on X and Y chromosomes. These are called hemizygous genotypes. Females, on the other hand, have two alleles for the genes on the X chromosome, so they can have either homozygous or heterozygous genotypes. This causes X-linked inheritance patterns to behave in slightly different ways between men and women. X-linked dominant diseases, like Fragile X syndrome, are caused by a mutation in the dominant allele of the X chromosome. Both heterozygous, X big D, X little d, and homozygous, X big D females have the disease. And males who are X big D, Y also have the disease, since you only need one mutant X chromosome to be present. But the chance of passing on the disease differs in these three situations. If a male has Fragile X, his genotype is X big D, Y, and his partner is female without the disease, then her genotype is X little d, X little d. Using a Punnett square, the father can make gametes which carry the mutant X big D allele, or gametes which carry the Y chromosome. And the mum only makes gametes carrying a normal X little d allele. Daughters of this couple have a 100% chance of ending up heterozygous, X big D, X little d, so they'd all have the disease. Sons, however, are in luck, because they get a normal X little d chromosome from the mum and a Y from the dad, and as a result they won't have the disease. On the other hand, if mum is heterozygous, X big D, X little d, and therefore has Fragile X, then she can make either normal X little d or mutant X big D gametes. And if her partner is a male without the disease, then his genotype is X little d, Y, and he can make normal X little d gametes or normal Y gametes. In this case, the sons have a 50% chance of ending up X big D, Y, and the daughters have a 50% chance of ending up X big D, X little d. So both sons and daughters have the same 50% chance of having the disease. In a pedigree, the pattern would show affected members in every generation, with females affected twice as often as males. Also, males only pass on the mutant allele to their daughters, which differentiates this pattern from autosomal dominant inheritance, while females pass on the mutant allele to both sons and daughters equally. Now, there's also X-linked recessive diseases like haemophilia, which develops when there are two recessive mutations on the X chromosomes. In this case, only homozygous recessive females, X little r, are affected, while heterozygous females, X little r, X big R, are carriers, and homozygous dominant females, X big R, are healthy. Males, however, can only be healthy X big R, Y, or affected X little r, Y. In other words, they can't be carriers. The chance of passing on the disease is also different for both males and females. Let's look at an affected male, X little r, Y, and a healthy female, X big R, In a Punnett square, the father can make mutant X little r gametes, or normal Y gametes, while the mother can only make X big R gametes. So the daughters have a 100% chance of being heterozygous, X big R, and so are carriers, and the sons have a 100% chance of being hemizygous, dominant, X big R, Y, so they would neither develop nor be carriers of haemophilia. Now let's say that the mother is a carrier, X big R, X little r, and the father is healthy, X big R, Y. The mother can make either normal X big R or mutant X little r female gametes, while the father can only make normal Y gametes, or dominant X big R gametes. So their sons would have a 50% chance of ending up hemizygous dominant X big R, Y, and a 50% chance of ending up hemizygous recessive and having the disease. But the daughters have 50% chance of ending up heterozygous X big R, X little r carriers, and a 50% chance of being X big R, homozygous dominant, which means they're healthy. In a pedigree, there would be twice as many affected males than females, the disease wouldn't transmit from father to son, and both mothers and daughters of an affected male would be definite carriers. Finally, let's look at Y-linked diseases, like some cases of baldness which only affect males. Because there's only one Y chromosome, a mutation on it results in the disease. The disease would always be passed on from father to son and never from father to daughter. In a pedigree, there would be only males affected, and the disease would be present in every generation. So, to recap, inheritance patterns can be visualized in pedigrees. With dominant inheritance, you only need one mutant allele to develop the disease, whereas in recessive inheritance, you need two mutant alleles to develop the disease. Now, autosomal inheritance means that the mutant allele is located in somatic chromosomes, while sex-linked inheritance, X-linked and Y-linked, means that the mutant allele is located in sex chromosomes. Finally, there are other more rare patterns of inheritance, such as mitochondrial inheritance, where females pass on mutations in the mitochondrial DNA to both sons and daughters, but males don't pass on the mutation.